Hello and welcome back to our immunology lecture series. As we have learned in the last video, there are two cell types that actively deal with cells during infection. Those are natural killer cells that recognize infected cells, so they are dealing with host cells. And there are also phagocytic cells, including macrophages and dendritic cells, that deal with microbes that have entered the system. So, how do phagocytic cells recognize pathogens? And again, phagocytic cells are macrophages, the big eaters, and dendritic cells. Those cells are able to recognize bacteria via a set of very specific surface receptors. And those receptors are referred to as pattern recognition receptors or toll-like receptors. And I'm quite passionate about those receptors because this is something I'm actively working on research-wise in my laboratory. On immune system cells, there is a wide range of those toll-like receptors present, and all of those toll-like receptors are in charge of recognition of different type of pathogen-derived molecules. Those molecules include bacterial lipoproteins. Other TLRs are able to recognize CpG-rich DNA fragment, which is not that common in eukaryotic cells, but quite common in bacteria. Other TLRs or pattern recognition receptors recognize short RNAs that are not common in human cells. And we also do have receptors that are able to sense lipopolysaccharides. And this is something quite specific for bacteria. This type of polysaccharides is not present in eukaryotes. So whenever those substances are sensed by the macrophages and dendritic cells via those receptors present on the surface, this leads to activation of a common downstream signaling pathway referred to as nuclear factor kappa B pathway. This pathway is fairly complicated, but also crucial for the function of the immune system. And again, for the exam, you don't need to memorize all of those different mechanisms that are triggered by activation of this transcription factor, but you need to keep in mind that this is the crucial player in activation of the immune system because it regulates expression of pro-inflammatory genes, and this is something quite important for activation of macrophages, but also neutrophils, dendritic cells, and other cells within the immune system. What you should remember, though, is that NF-kappa B triggers release of cytokines, and cytokines are the main molecules involved in mediation of inflammation. Cytokines are regulatory proteins secreted by all white blood cells. Those guides bind to specific receptors on target cells, triggering signal transduction pathways and altering the gene expression patterns. So once cytokines are released by immune system cells, this leads to activation of further cells within the immune system, and this is sort of a feed-forward loop. Cytokines and the receptors exhibit very high affinity for each other. So cytokine signaling between immune system cells is quite specific. Moreover, cytokines can be either pro or anti-inflammatory, and the pro-inflammatory cytokines are interferon gamma, tumor necrosis factor alpha, which is the most important and also prototypic pro-inflammatory cytokine, interleukin 2, 6 and 12. This is of course not the full list, and there are many more cytokines that are known to be pro-inflammatory, but you don't need to remember the details. However, keep in mind that the most important one is tumor necrosis factor alpha. In addition to those, there are also anti-inflammatory cytokines that suppress the immune system, including interleukin 4, 5, 10, and TGF-beta. So, how do interferons affect the immune system? Interferons are actually extremely important in letting the system know that there is a viral infection going on. And this has been recognized as one of the very crucial points in COVID-19 at the moment. Interferon response and subsequent release of cytokines actually leads to many, many complications in the COVID-19 infection. Both in simple terms, if a virus infects a cell, this leads to release of interferons, and those interferons activate natural killer cells that then in turn kill the infected cells. So, in terms of pro-inflammatory cytokines, again, the most important one and the only one you need really to remember is tumor necrosis factor alpha. This is the most important mediator of inflammation, and you will hear a lot about this molecule later on as well. And indeed, tumor necrosis factor alpha is also a target of many therapies against autoimmune disorders. You can use neutralizing antibodies and also molecules that block the receptor for this cytokine. So this is something that you should certainly remember. So, what happens if TNF can bind to a receptor? This actually triggers, as usual, NF-kappa-B response. But NF-kappa-B response, mediated by TNF-alpha, is very strong. So the level of inflammation that you can actually end up with is fairly strong. So why is actually inflammation important? And why does inflammation play a role in an immune system? Inflammation manifests in 5 hallmarks, and this is something we need to keep in mind in order to understand the role of inflammation in the innate immune system. The first sign of inflammation is redness. The second hallmark of inflammation is pain. Third is heat, and this is already part of the immune system. Development of heat changes the temperature in the tissue to a temperature range which is less favorable for the microorganisms. Then we have swelling, and last but not least, loss of function of the organs. So how does inflammation affect the whole system? So if we have a tissue injury caused by a physical or chemical agent or pathogenic microorganism, this leads to capillary widening, increased capillary permeability, and attraction of white blood cells. And this is actually the most important step if it comes to innate immune system. Inflammation actively activates macrophages and allow them to migrate. Afterwards, we have the systemic response, which induces fever. And again, fever is actually beneficial if it comes to infection in short term, because the organism is shifting the temperature to a temperature range which is less favorable for the infecting microorganism. So, let me sum up what we have learned about the innate immunity. And again, you will hear much more about the immune system in part 3 in a lecture series delivered by Dr. Edwards. But for now, what you need to remember when it comes to innate immunity is that there are 2 types of immunity, innate and adaptive. You need to keep in mind that innate immunity relies on mechanisms already existing before microbe infect hosts. Innate immunity is the first and second line of defense, and it consists of physical, chemical, but also cellular mechanisms. It relies on non-specific mechanisms, has no memory for subsequent exposure, and activation of the immune system response induces secretion of cytokines and leads to inflammation. With this, I would like to close our video series on the innate immune system, and I'll see you next week, where we're going to be discussing the adaptive immune system, and also where we're going to be looking at the principles of vaccination, autoimmunity, and also immunosuppression. Thank you very much for your attention.